And now it is my privilege to introduce our first presenter for today's session, Sean Morrison. Professor Sean Morrison is the director of the Children's Medical Center Research Institute at UT Southwestern, and he's the Mary McDermott Cook Chair in Pediatric Genetics, as well as an investigator of the Howard Hughes Medical Institute. He made critical contributions to the field, discovering new markers to prospectively identify, isolate, and characterize hematopoietic stem cells. He also made considerable advance in our understanding of how HSCs are regulated by the bone marrow perivascular niche. And I will now turn the presentation over to Sean.
Thanks, Christina. Thanks to ISEH for the opportunity to participate in this with Simone. So I'll spend the next 20 minutes just summarizing our data on the nature of the niche for hematopoietic stem cells in the bone marrow, where, of course, the key question is which cells actually secrete the factors under physiological conditions in vivo that are required to maintain hematopoietic stem cells in the bone marrow. I'll also address the relationship between the hematopoietic stem cell niche and skeletal stem cells, and then just briefly mention some of our new and unpublished data related to new growth factors in the bone marrow. So everybody knows that the bone marrow has got lots of hematopoietic cells, but it's also got a complex vascular network and a complex network of stromal cells within the bone marrow. So on the right-hand side of the screen, there are sinusoidal blood vessels that are shown here in purple. There are arterioles that are shown here in green. And there are transition zone vessels which hook up the arterioles that bring in arterial blood to the sinusoids, which carry the blood back out of the bone marrow. The transition zone vessels are referred to as such because they have properties that are intermediate. Because they hook the sinusoids to the arterioles, their properties are intermediate and are difficult to characterize either as sinusoids or as arterioles. So there have been a lot of different bone marrow cells that have been suggested to be part of the hematopoietic stem cell niche. But prior to the data that I'm going to tell you about this morning, nobody had done two kinds of direct experiments. One direct experiment that I'm going to tell you about is a systematic examination of the expression patterns of critical niche cell factors in the bone marrow so that we can systematically examine what are all of the cells that express these factors, which cells express them at high levels, which cells express them at low levels, which cells don't express them at all. And then secondly, nobody had ever conditionally deleted specific niche cell factors from candidate niche cells. And so there have been a lot of different models out there in terms of which cells were secreting the factors that promote stem cell maintenance, but the models, including the ones that we proposed initially, hadn't been tested directly functionally. So our interest in this area started more than 10 years ago when we discovered that SLAM family markers could be used to highly purify hematopoietic stem cells with a simple combination of markers. So for a long time, for 20 years prior to that, it had been possible to highly enrich hematopoietic stem cells by flow cytometry using markers that have been identified mainly by the Weissman lab. And the difficulty was that it needed about 12 different antibodies and 5-color flow cytometry to purify the stem cells. And that was too complicated a combination of markers to cut sections through hematopoietic tissues and to know where cells that were likely to be stem cells were actually localized. But with SLAM family markers, we could do, for the first time, a simple 2-color stain with 3 different antibodies and see cells that were very likely to be stem cells and where they localized. And when we did that, we found that, like this cell that's shown in this image, most of the cells that we found, most of the hematopoietic stem cells we found, were immediately adjacent to sinusoidal blood vessels, both in the bone marrow and the spleen. So this white stain is a pan-endothelial antigen. This is the lumen of the sinusoid. This giant yellow cell is a megakaryocyte. Megakaryocytes are also well known to localize to sinusoids throughout the bone marrow. So when we saw that, we proposed that there was a perivascular niche for the maintenance of hematopoietic stem cells. But to the point that I made on the last slide, at the time we didn't know anything about which cells actually secreted the factors that promoted stem cell maintenance. We assumed that at least some of those cells were around sinusoids throughout the bone marrow, but that wasn't tested directly. So we decided to directly test our model, and while we were at it, all of the models that have been proposed by anyone about the nature of the niche, initially by knocking GFP or DS-RED into two known niche cell factors, stem cell factor and CXCL12. And in an effort to disprove our model, we also knocked GFP into the angiopoietin-1 locus, which had been proposed to be an osteoblast-specific mechanism for the maintenance of hematopoietic stem cells. So we used those reporter alleles to systematically examine the expression patterns of all of these factors throughout the bone marrow. Then we also made floxed alleles of each of these factors, and we identified Cre alleles that were expressed in every candidate niche cell population that anyone had proposed. And we mated all of the Cre alleles with all of the floxed alleles to systematically conditionally delete each of these factors in all the candidate niche cell populations to assess which ones were actually functionally important as a source of factors for stem cell maintenance. So this is what we saw when we knocked GFP into the stem cell factor locus. So this is a low magnification image of a femur where this is bone on the top and bone on the bottom, and this is all bone marrow in the middle. So if you look at these images with high magnification, the osteoblasts that live here at the interface of bone and bone marrow, they don't express stem cell factor at all. In fact, if you look, you see these little rings of green stem cell factor expression that co-localize with red endothelial staining throughout the bone marrow. So all these rings, these are the sinusoids that are present throughout the bone marrow. And the vast majority of SCF expression is around the sinusoids, where we had seen most of the hematopoietic stem cells. These blood vessels here are arterioles. We also see some SCF expression around the arterioles, but less than what we see around the sinusoids. So if you look at high magnification, there's a very low level of SCF expression in the endothelial cells themselves. Most of the SCF expression is in perivascular stromal cells that surround the sinusoids. That is, they're on the abluminal side of the endothelial cells. So initially we had no idea what these cells were because at the time there had been very little characterization of perivascular stromal cells in the bone marrow. But we used SCF as a marker to isolate the cells, and we did gene expression profiling, and we found from the expression profiling that we could identify these cells based on their expression of full-length leptin receptor. So we confirmed the utility of leptin receptor as a marker to identify those cells by mating leptin receptor Cre with a conditional reporter. And when we did that, we got recombination of the reporter specifically in these perivascular stromal cells, not in endothelial cells themselves, not in hematopoietic cells in the bone marrow, not in osteoblasts at the interface of bone marrow and bone. So this gave us a molecular handle on these cells and allowed us to study their properties in vivo, and most importantly, to conditionally delete specific niche cell factors from these cells to see if we would actually get stem cell depletion. Before I tell you about those functional experiments, we also knocked DS-RED into the CXCL12 locus, and to a first approximation, we see the same expression pattern for CXCL12 as we had for SCF. That is, we see these rings of red CXCL12 expression that are around sinusoids throughout the bone marrow. So the cells that express SCF and CXCL12 in the bone marrow at a high level are overwhelmingly these leptin receptor positive perivascular stromal cells. We see very low levels of SCF and CXCL12 in endothelial cells. Osteoblasts don't express detectable SCF, but they do express a very low level of CXCL12, about 1,000-fold lower than what we see in the leptin receptor positive cells and about 10-fold lower than what we see in the endothelial cells. The vast majority of these leptin receptor positive cells are around sinusoids throughout the bone marrow, but they are also around arterioles, particularly around small diameter arterioles in the bone marrow. So to systematically, in an unbiased way, examine the expression patterns of SCF and CXCL12, we did the following experiments. where we recombined TD tomato with leptin receptor Cre to identify all of the leptin receptor positive stromal cells in the bone marrow. And then we gated on those cells and found that 99% of those cells express a high level of SCF-GFP. Similarly, we recombined in the leptin receptor positive cells and then looked at what fraction of them express a high level of CXCL12. And again, 99% of these cells express a high level of CXCL12. So what if we do the experiment the other way around? If we gate on all of the stromal cells that express a high level of SCF-GFP, so now this gate doesn't include the endothelial cells, which express 100-fold lower levels of SCF. But if we just look at all of the cells that express a very high level of SCF, about 95% of them are recombined with leptin receptor Cre. So there continue to be a lot of suggestions about different cell types in the bone marrow that are key sources of niche cell factors, and there will be other cells that are important sources of factors that regulate hematopoietic stem cell maintenance. But in terms of the known canonical niche cell markers, there's not a lot of room here for distinct cell populations that are a major source of SCF. Similarly, if you look at CXCL12, about 90% of these cells are in the leptin receptor positive population. So we conditionally deleted these factors, SCF, CXCL12, or angiopoietin-1. This is just a subset of the Cre alleles that we used that were most informative. To make a long story short, with angiopoietin-1, it turns out not to be expressed by osteoblasts at all, and it's not required for hematopoietic stem cell maintenance or hematopoietic stem cell quiescence. In fact, hematopoietic stem cells themselves express the highest levels of angiopoietin-1, and they secrete angiopoietin-1 as a mechanism to regulate the regeneration of the vascular niche after injury. So if we focus on SCF and CXCL12, if we delete these factors ubiquitously, we see hematopoietic stem cell depletion as you would expect. Consistent with the inability to detect SCF expression in osteoblasts, there's no effect on HSCs or hematopoiesis when we delete SCF in osteoblasts. As I mentioned to you before, we see a very low level of CXCL12 expression in osteoblasts. And when we delete it in osteoblasts, there's no effect on hematopoietic stem cells, but we do see a depletion of early lymphoid progenitors. And we do see a subset of early lymphoid progenitors that localize to the endosteum. So we think that there is an osteoblastic niche, but we don't think it's a niche for stem cells. We think it's a niche for a subset of early lymphoid progenitors. Consistent with the inability to detect these factors in hematopoietic cells, we get no phenotype when we delete them in hematopoietic cells. Now there are three markers that have been proposed for periarteriolar stromal cells. We've mated each of these Cre alleles with conditional reporters, and we've mated each of them with either SCF-GFP or CXCL12-DS-Red. We're not able to detect SCF expression or CXCL12 expression in any of these periarteriolar cell populations. And when we conditionally delete SCF or CXCL12 in any of these periarteriolar cell populations, there's no effect on hematopoietic stem cells and there's no effect on hematopoiesis. Similarly, we don't detect SCF or CXCL12 in megakaryocytes, and we delete in megakaryocytes, there's no effect on stem cells or hematopoiesis. In contrast, when we delete in endothelial cells or in leptin receptor positive cells, now we do see hematopoietic stem cell depletion consistent with the expression patterns of these factors that we had seen, suggesting that there are two cell types that are important sources for at least these known stem cell factors, endothelial cells and leptin receptor positive cells, and predicting that if we deleted these factors in both endothelial cells and leptin receptor positive cells in the same mice, that we should see a profound depletion of hematopoietic stem cells. So we tested that by generating mice that carry both Tie2 Cre and leptin receptor Cre, and consistent with the prediction, we do see a profound depletion of hematopoietic stem cells, either based on marker expression or based on function and transplantation assays. In fact, if we try to age these mice, they actually die of hematopoietic failure because they have no quiescent and no serially transplantable stem cells that are left in the bone marrow of these mice. So that provides direct evidence then that quiescent and serially transplantable hematopoietic stem cells depend on this perivascular niche for their maintenance. The other factor that's been limiting in this field is the rarity of hematopoietic stem cells themselves. When we proposed in 2005 that there was a perivascular niche for hematopoietic stem cell maintenance, our conclusions about stem cell localization, I think, were based on the analysis of 36 hematopoietic stem cells that we found over a period of weeks in sections through long bones. So, you know, what you'd really like is to be able to see the whole picture, where we could look at large segments of bone marrow, know where all of the stem cells are, and instead of basing our conclusions on dozens of hematopoietic stem cells, based on our conclusions on hundreds or thousands of hematopoietic stem cells. So there were a number of technical advances that were required to get there. One was the identification of a new genetic marker for hematopoietic stem cells called alpha-cotulin. This helped us to get away from a reliance on antibodies to identify the cells in thick segments of bone marrow. So alpha-cotulin, you've probably never heard of it. There's very few papers on it. It's been proposed to be a cytoskeletal linker protein, but we can't find any function when we knock it out in the hematopoietic system. It is a great marker, though, for hematopoietic stem cells because we knock GFP into the alpha-cotulin locus. Only 0.02% of bone marrow cells are GFP positive. Now, all of the hematopoietic stem cell activity falls within this fraction of alpha-cotulin positive cells in the bone marrow, but this is not a pure population by itself. In fact, if you look over at the functional data on the right, this is all limit dilution reconstitution data. On average, in these experiments, 1 in 37,000 bone marrow cells gave a long-term multilineage reconstitution upon transplantation into irradiated mice. If we just sort based on GFP and transplant single cells, 1 in 7 single cells gives long-term multilineage reconstitution. There's no activity or virtually no activity in the alpha-cotulin negative bone marrow fraction. But we can further increase the purity of the alpha-cotulin positive cells by also including c-KIT as a marker. When we do that, it's about 30% of the single cells that we transplant that give long-term multilineage reconstitution. Pretty similar in purity to when you isolate stem cells with SLAM family markers or other good markers. So it's about half of these alpha-cotulin positive cells that stain with c-KIT. These cells are uniformly positive for SLAM family markers, and so we have identified the localization of these alpha-cotulin positive, c-KIT positive cells through the bone marrow. So that was, our ability to do that was enhanced by also implementing methods to clear bones and bone marrow. So you can see that after the clearing protocol, we can actually see through the bones and read text looking through the bones. And this makes it possible to do high throughput confocal imaging of thick segments of bone and bone marrow, where we can pick out all of the alpha-cotulin positive and c-KIT positive cells throughout the bone marrow, and then computationally assess their positions relative to any landmark we want, whether it's a bone surface or a sinusoidal blood vessel or an arteriole. So just to illustrate, so essentially with these images, we're able to make three-dimensional reconstructions, digital reconstructions of the bone marrow, where we can fly through and do close-ups on individual hematopoietic stem cells and look at their positions relative to other cell types. So just to illustrate that, I'll show you a short video where the bony-looking stuff is bone, the stuff in the middle is bone marrow. Alpha-cotulin among hematopoietic cells is highly restricted to hematopoietic stem cells, as I've shown you. But it is also expressed in sinusoidal endothelial cells. So you can see here this blood vessel-looking thing in green through the bone marrow. In fact, that's a sinusoid, a large sinusoid that collects the venous circulation from small sinusoids. It's green because the endothelial cells that create the wall of the sinusoid are alpha-cotulin positive. But we can distinguish the sinusoidal endothelial cells, which are green, from stem cells because the stem cells also express c-KIT. So endothelial cells are green, stem cells are green and white, and niche cells are red in this video. So we can fly in and do close-ups on individual stem cells and look at their interaction with the cells that we'd identified genetically as important sources of niche factors. So let me just draw your attention to this cell. This cell is positive for alpha-ketulin and c-KIT, so it's likely to be a stem cell based on the markers that expresses. This is typical of what we see in these images. This is a regular sinusoid that drains into that large sinusoid that I pointed out when we looked. The vast majority of hematopoietic stem cells that we see are like this one, immediately adjacent to sinusoidal blood vessels, consistent with what we had concluded based on staining with SLAM family markers in 2005. You'll also see that these cells are very consistently and closely associated with both endothelial cells, shown here in green, and leptin receptor positive cells, shown here in red. So the imaging is consistent with the genetics in the sense that these are the two cell types that we know are genetically important to secrete factors for stem cell maintenance, and the stem cells are very consistently closely associated with those markers. The leptin receptor positive cells represent about 0.3% of cells in the bone marrow, and they are throughout the entire bone marrow. They have long processes, so in these kinds of thick images, they look more frequent than they actually are. So by looking at literally thousands of hematopoietic stem cells, we see that in the diaphysis of bones, less than 1% of hematopoietic stem cells are close to bone surfaces. There are more, about 7% of stem cells are close to bone surfaces in the metaphysis where the trabecular bone is located. If we look at their localization relative to blood vessels, around 80% of hematopoietic stem cells, irrespective of whether we're looking at dividing hematopoietic stem cells or non-dividing hematopoietic stem cells, are most closely associated with sinusoidal blood vessels. We see another 10% of dividing and non-dividing hematopoietic stem cells that are more closely associated with arterioles, and there's another 10% of the cells that are most closely associated with the transition zone vessels. So just to sum this up, there's a perivascular niche for hematopoietic stem cells in the bone marrow that's created mainly by leptin receptor positive perivascular stromal cells and endothelial cells that are the key sources of factors for hematopoietic stem cell maintenance. So Paolo Bianco initially, and then Simon Mendez-Ferrer and Paul Fournette published that skeletal stem cells were a component of the niche for hematopoietic stem cells. And so it was natural for us, having identified cells that we knew were an important source of factors for stem cell maintenance, to test their idea. And what I'm going to show you now is that their conclusion is correct, that the cells that secrete factors for hematopoietic stem cells do include the skeletal stem cells that are an important source of bone for the maintenance of the adult skeleton. So we started that work by just doing, again, the unbiased experiment of taking a variety of Cre alleles that have been suggested to mark mesenchymal stem cells, and then recombining a conditional reporter, and then taking all of the bone marrow cells in an unbiased way and plating them out in culture, and then asking what percentage of the fibroblast colonies that formed in culture were actually marked with recombination by the Cre allele. When we do that with, so fibroblast colonies, as I'm sure everybody aware, are associated with skeletal or mesenchymal stem cell activity. It's those colonies which, when passaged, give rise to multipotent osteogenic, adipogenic, and chondrogenic progenitors from the bone marrow. So PRX1 Cre, consistent with what others had published, marks all of the fibroblast colony-forming cells in the bone marrow. Leptin receptor Cre marks virtually all of the fibroblast colony-forming cells in the bone marrow, about 94% of them. We were unable to find any marking with Nestin Cre-ER or NG2 Cre-ER or neural crest markers when we looked at the adult bone marrow. So single leptin receptor positive cells form fibroblast colonies in culture that undergo multilineage differentiation into bone cells and fat cells and chondrocytes. Now, it's important to note, though, that these leptin receptor positive cells, they are heterogeneous. They're not heterogeneous in terms of SCF expression. They're not heterogeneous in terms of CXCL12 expression, but they are heterogeneous in terms of their capacity to form mesenchymal colonies. Our best guess right now is that it's about 10% of these leptin receptor positive cells that are multipotent skeletal stem cells. So leptin receptor gave us a marker that allowed us to assess the physiological function of these cells in vivo. We could do that by mating leptin receptor Cre with a conditional reporter, and in this case also with a reporter of osteoblast identity. And when we looked in 2-month-old mice, we see very little contribution of bone. But if we age the mice, now we see a large contribution to the osteoblasts that maintain the adult skeleton. And if we quantify these effects, we only see about 5% of osteoblasts that derive from the leptin receptor positive cells in 2-month-old mice. But it goes up as the mice age, and by 14 months of age, most of the osteoblasts derive from these leptin receptor positive cells. So that tells us that the leptin receptor positive cells are not responsible for the formation of the skeleton during development. They're responsible for the maintenance of the skeleton throughout adulthood. They not only maintain the normal, they not only give rise to most of the osteoblasts that maintain the normal turnover of bone in the skeleton, but they also are the main cell type that's responsible for fracture healing when the bones break. They are also the main source of adipocytes that accumulate with age within the bone marrow. Adipocytes at all ages arise overwhelmingly from these leptin receptor positive cells. Cartilage forms during development, and it doesn't turn over during adulthood. I mean the regular articular cartilage in joints. As a result, the leptin receptor positive cells don't form the cartilage that occurs during development. But if you injure bones, so you form cartilage normally around a fracture site, most of this cartilage that forms around a fracture site does arise from the leptin receptor positive cells. So they're not the normal source of bone that arises developmentally, but they are the normal source of cartilage that arises developmentally, but they are the major source of cartilage that fixes injuries. So just to sum up then what I've presented, the hematopoietic stem cell niche is, at least in terms of the key sources of known canonical niche factors, there will be other cell types that are involved in the niche. Leptin receptor positive cells and endothelial cells are the sources, are key sources of SCF and CXCL12 for hematopoietic stem cell maintenance. Most of the non-dividing hematopoietic stem cells are immediately adjacent to sinusoidal blood vessels throughout the bone marrow. There's another 10 to 20% of hematopoietic stem cells that are adjacent to arterioles in the bone marrow, just depending on how generous you want to be and what you count as an arteriole. The leptin receptor positive cells also include the skeletal stem cells that are the major source of bone and adipocytes in the bone marrow. And I won't take you through this data in the interest of time, but we know that the skeletal stem cells are regulated by leptin, that leptin receptor is acting as a nutritional sensor on these cells, and that leptin that's stimulated by increased dietary intake or adiposity acts on the skeletal stem cells to promote adipocyte formation at the expense of bone. There are also other growth factors that are expressed in the bone marrow by these leptin receptor positive cells that have uncharacterized growth factor activities that we're in the process of studying now. So I'll just finish up then by saying that all of the experiments that I've told you about in the bone marrow, we've also done the same experiments in the spleen. Hematopoietic stem cells in spleens with extramedullary hematopoiesis are also found overwhelmingly, if not exclusively, around sinusoids in the red pulp of the spleen, where it's again endothelial cells and perivascular stromal cells that secrete the factors that promote hematopoietic stem cell maintenance and extramedullary hematopoiesis. Although the markers that these endothelial cells and perivascular stromal cells express in the spleen are different from in the bone marrow, and so it looks like the identity of those cells is a little bit different. So there's been a lot of people in my laboratory that have contributed to these studies. Most notably, Lei Ding originally made all the mice that were the basis for the experiments in the initial part of the talk. He's now got his own lab at Columbia University. Bo Zhou discovered that the leptin receptor positive cells have mesenchymal stem cell or skeletal stem cell activity within the bone marrow. He's also about to go off and start his own laboratory. And Ray Yu showed that those cells were regulated by leptin. So I'll stop there and turn it over to Simone.
Okay, so just as a reminder for everybody who's been listening, we will do all question and answers at the end. But if you have questions already, please start typing them into the chat window so that we can start reading them and passing them through the speakers, and we will be more efficient with the discussion at the end. So now it is also my privilege to introduce Dr. Simon Mendes-Ferrer, who is a group leader at the Wellcome Trust Medical Research Council Cambridge Stem Cell Institute and the National Health Service Blood and Transplant, and he's also a reader at the University of Cambridge, UK. Simon discovered a new link between hematopoietic stem cells and the nervous system and how these cells follow the circadian rhythms of the body. His work has contributed to the understanding of the role of the hematopoietic stem cell niches in homeostasis and also in the development of neuroproliferative disorders. And so I will now turn the presentation over to Simon.
Thank you very much, Christina, and the organizers for this opportunity to participate in this summer webinar with Sean. So our views on the hematopoietic stem cell niche has significantly evolved over the past few years. And maybe one key emerging concept is the shift from the probably oversimplified view that there is one agency and one niche for it to the realization that there may be several agencies, or at least several agency states, and each of them might be regulated by different types of niche cells. Therefore, finding heterogeneity among agencies has been paralleled by the discovery that different types of interconnected cell types regulate agency function. So traditionally, two main types of niches were proposed, endothelial niches in opposition to bone and vascular niches farther from the bone. But however, we have learned from Sean and from work from others as well that most HSCs reside near blood vessels in the bone marrow. However, we also know that this traditional dichotomy between endothelial and vascular niches is probably not valid because the endothelium is highly vascularized. At the same time, as Sean described elegantly, there are different types of blood vessels in the bone marrow. While 85% of HSCs, according to their work, reside near sinusoids, it's important to note that also sinusoids take over 90% of the bone marrow space. And it's still possible that the remaining 15% HSCs might be also located near endothelial vessels, such as transition vessels or arterioles, and somehow even they might be also different subpopulations of HSCs, which remains to be investigated. So these different types of blood vessels, thanks to the development of different markers by different labs, such as VEGF receptor 3, demonstrated by Shaheen Rafi to label these sinusoidal endothelial cells, or endomucin high levels showed by Ralph Adams to mark this endothelial transition zone vessels. This anatomical separation of these different types of blood vessels is now possible, and to study populations associated with them is also possible. I'd like to highlight that probably, despite some discrepancies in the field that needs to be resolved, there is also quite a few take-home consensus messages, namely that endothelial cells and bone marrow mesenchymal progenitor and stem cells, this can be labeled through a different combination of markers, which seem to mark partially overlapping populations, such as nestin GFP, as Paul Fournette and we showed, or leptin receptor, or PRX1 trace cells, which together with endothelial cells, as shown elegantly demonstrated, are important sources of HSC niche factors. At the same time, a number of future challenges appear ahead, and some of them are just mentioned here. Are there specific subpopulation of endothelial cells and perivascular mesenchymal stromal cells with specialized niche functions? Are there also specific endothelial cell pericyte interactions which will be required for some niche functions? And do different niches imprint different HSC states? So addressing these questions has not been very trivial, because in general, deleting single molecules in single niche cells has not always given a phenotype, or has not been a robust approach due to redundancy, given the fact that most niche factors are produced by different cell types, to compensation potentially from other cell types, which can act regulate a factor when you delete it in a very specific cell type, or due to inefficient recombination. In some cases, and this is, for instance, the case for nesting kriar mice, you just target tissue cells to have a strong phenotype. So in general, the strongest HSC phenotypes have been observed after deleting candidate niche factors using constitutive krillines. However, one has to bear in mind that some of these effects were affecting developmental hematopoiesis before the bone marrow stage, and also that some of these constitutive krillines target multiple types of stromal cells in the bone marrow, and therefore the combined deletion of the factors in multiple types of different cells might give per se a stronger phenotype than specific deletion in very narrow cell populations. However, the specific identity of the niche populations having a direct impact on hematopoietic stem cells is, to my understanding, is still unresolved. So it's also possible that the overlap of niche cell populations might be actually higher than previously anticipated, and this is because cell populations that have been characterized in great detail often represent only a subset of the populations that have been targeted with candidate krillines having a hematopoietic phenotype. Therefore, the overlap among different krillines may be larger than previously anticipated. And then there is also the additional complexity that the mesenchymal stromal cells may be largely heterogeneous, as hematopoietic stem cells have been shown to be. And our own data shows functional evidence that of functional segregation of different types of stromal cells in the perinatal bone marrow. For instance, during perinatal development in the bone marrow, we found that nesting GFP negative cells had a primary function in giving rise to cartilage and bone formation, and this has been reproduced by the groups of Paul Frenet and Cronenberg and Team 1. And in contrast, the nesting GFP positive mesenchymal stem cells showed very little contribution to skeletal formation, but instead had a primary function of contributing to establish the niche robotic stem cells in the developing bone marrow. So this heterogeneity of the stromal cells also is unspecific functions. It's emphasized by, for instance, deletion of CXCL12 using nesting KRT2, which at this stage overlaps with nesting GFP, not so much at later stages, when also the recombination efficiency of this is extremely low. However, by deleting CXCL12 only in this narrow population, there was a problem in the colonization of the bone marrow by circulating HSPCs, when the factor was specifically deleted at the time when HSPCs migrate from the fetal liver to the bone marrow. And similarly, this heterogeneity is further emphasized for potentially different origins for MSCs in the bone marrow at different stages of life. And we show that in the perinatal bone marrow, some of these nesting positive cells are actually derived from the neural crest, and also that preventing the migration of these neural crest derivatives to the bone marrow, there was also a problem in the colonization of the bone marrow by circulating hematopoietic stem cells. And a number of different labs have also found different ways of bone marrow mesenchymal stem and progenitor cells that might exhibit different functions throughout life. I already mentioned some contribution from the neural crest, mainly during the fetal period. Other groups have shown that different mesenchymal progenitors are specialized in giving rise to bone and stroma formation, and these progenitors intriguingly exhibit different markers at different stages of life, raising the possibility whether there are new populations coming into place or just changing markers over time. For instance, in the neonatal period, Team 1 showed that Gremlin-1 labels these skeletal stem cells that actually give rise to most of the skeletal lineages, and Sean just mentioned how leptin receptor Q derivatives actually have this role in adult life, and very differently from nesting KRT2 targeted cells, which very scarcely contribute to skeletal formation. Also, it has become apparent that maybe more differentiated mesenchymal progenitors might actually participate in bone turnover for instance, work from David Cullen's group showing that MX13 labels restricted osteolineage cells, probably having a specialized role in these bone turnover. So we were interested in the relationship between these nestin positive cells, stromal cells, with other components of the nervous system also derived from neural crest, such as sympathetic neurons, shown by the Fulfronet's group to regulate mobilization of HSCs, and also Schwann cells that cover these sympathetic neurons, and suggested by Nakauchi's group to be a main source of TGF-beta required for HSC quiescence. So we detected that sympathetic nerve fibers remain closely associated with these nestin GFP-positive cells in adult bone marrow. And since the sympathetic nervous system is a stress response system, we decided to have a look at the role, the relevance of this regulatory network in a stress situation. And we decided to look at myeloproliferative neoplasms because these are cancer stem cell derived diseases originated by mutations in hematopoietic stem cells, but they impose a stress situation and inflammatory stress in the microenvironment, and they lead to the fibrosis and osteosclerosis of the bone marrow, suggesting that the mesenchyme might play an important role in the pathogenesis of this disorder. And we realized that actually these sympathetic nerve terminals and the associated Schwann cells were extremely sensitive to the inflammatory cytokines derived from the mutated hematopoietic cells. And that this neuropathy that the mutated cells cause in the bone marrow actually preceded and sensitized these nestin-positive stromal cells to undergo cell death. So we thought that somehow the neuropathy might sensitize the stromal population in the inflammatory context of MPN, and we decided to intervene in this neuropathy by providing adrenergic agonists that would supply the source of the missing neurotransmitter and activate the stromal cells. And we decided to use beta-3 adrenergic agonists because they don't activate hematopoietic cells that we know, only stromal cells, and they were capable of rescuing these nestin GFP-positive cells from cell death. And intriguingly, this rescue was also associated with a blockade of disease in peripheral blood, particularly neutrophilia, was markedly blocked, and also the thrombocytosis was greatly improved. And because these dystrophic megakaryocytes are actually the source of cytokines that lead to the fibrosis and osteoporosis of the bone marrow, mice chronically treated with beta-3 agonists showed dramatic improvement of the fibrosis and the osteoporosis in the bone marrow. So to conclude this last part, we have shown that mutated hematopoietic stem cells damage their own niche in myeloproliferative neoplasms, and they can initiate disease only after overcoming the control by the niche. Therefore, we believe that protecting the niche in this set of diseases might be therapeutic and probably a good target in combination with treatment directly targeting the mutated cells. But more generally, from the previous part, more general conclusion, one has been already raised: endothelial cells and perivascular stromal cells can be marked with different markers, but are important HSC niche cells. But in the future, because the DMSs are heterogeneous in origins, markers, and functions, we need to devote effort to identifying particular populations that might be relevant in each case. So future work is needed to dissect these specific cell types involved, their interactions, particularly these endothelial cells with perivascular cells, and their specialized functions in HSC regulation. So I stop here just by acknowledging the people who did the work. Most of the culinary study has been developed by Andres Garcia, and Lorena Rand developed the MPN project in the past, and she has now her own lab. And these are our lab members and funding agencies. Thank you very much for your attention.
Thank you, Simon. Many thanks to Simon and to Sean as well. And we have now time for a few questions. I have one question already in the chat showing up here. It is a three-part question for Sean. One is, are leptin receptor positive cells the exclusive bone marrow source of the MSCs that can be grown in culture? Two, are the leptin receptor positive cells also present in other MSC containing tissues, for example fat? And three, is there any idea if leptin receptor is similarly informative for human?
Let me start by first emphasizing something that Simone said, that there's different waves, and then there's been a number of laboratories that have published evidence that support this idea. There are different waves of mesenchymal progenitors developmentally, neonatally, and during adulthood. So there are different cells that are forming the skeleton during adulthood, that are completing the development of the skeleton neonatally, and that are maintaining the skeleton during adulthood. And there may be, different mesenchymal progenitors that exist simultaneously, particularly neonatally within the bone marrow. And so that will all be interesting and important to work out. If we just restrict our focus per the question to the adult bone marrow, 94% of the CFUF activity in the adult bone marrow derives from the leptin receptor positive cells. If we fracture the bone marrow, we see the leptin receptor positive cells give rise to most of the cells that make an enduring contribution to bone repair, but not all of them. And it's known that there's also a periosteal bone progenitor population that participates in bone repair. And so that does play some role in the involvement of fracture repairs. From our data, it looks like mesenchymal stem, the leptin receptor positive cells are the main source of bone and adipocytes in adult bone marrow, but we wouldn't rule out the possibility that there would be other populations that make a small contribution. Other laboratories have looked at leptin receptor expression in human mesenchymal stem cells, and obviously it's not possible to do the direct experiment of fate mapping in humans that we've done in mice to persuade ourselves that the leptin receptor positive cells are the major source of bone. But if you just look in human mesenchymal stem cell cultures, there's about 4 laboratories now that have published that leptin receptor is expressed in those cells. And then in terms of other tissues, there are leptin receptor positive cells in other tissues that can form fibroblast colonies in culture, but we've not yet studied the physiological function of those cells. And it probably varies from tissue to tissue. And so we don't know to what extent the leptin receptor is a good marker for identifying cells that have CFUF activity in other tissues, or to what extent. Certainly in some tissues there will be other cell populations with fibroblast activity.
Thank you. If I might introduce one more question on these leptin receptor positive cells. Your 3D visualization of the leptin receptor positive cells and HSCs is really, really beautiful, and I appreciate how you said that there are fewer leptin receptor positive cells than it looks like because of the 3D reconstruction, but it still looks like there are more leptin receptor positive cells than HSCs. So my question is, what do you think localizes each HSCs next to the leptin receptor positive cells that it is next to? Do you think there could be something else that ultimately decides which one is the lucky cell that is going to host an HSC?
Yeah, it's a good question. There's multiple parts to the answer to that question. The frequency of the leptin receptor positive cells in the bone marrow is about 100 times higher than the frequency of hematopoietic stem cells. And, it's clear that certain treatments in the bone marrow can increase the frequency of hematopoietic stem cells, say about 10-fold, like after cyclophosphamide G-CSF treatment. And so we've always known that there has to be excess capacity within the niche, although it's a capacity that cells, based on the data we've seen so far, don't normally access. But we also know, for example, from Simone's work, that hematopoietic stem cells routinely mobilize into circulation from the bone marrow, even under normal physiological conditions, and then probably leave the circulation and rehome to a different bone marrow compartment. So we think the presence of these leptin receptor positive cells around sinusoids creates microenvironments that have the potential, at least, to sustain hematopoietic stem cells when they come out of the circulation. Two other points I think that are important to consider in light of that question. One is that although so these leptin receptor positive cells account for most of the SCF and CXCL12 that's made within the bone marrow because of their frequency and the level at which they express it. Hematopoietic stem cells are not the only cells that depend on CXCL12 and SCF, and I should say they also express other factors. So there are probably a variety of other hematopoietic progenitors in the bone marrow that also depend on these cells and that localize close to them. Secondly, the leptin receptor positive cells are essentially homogeneous for SCF expression and CXCL12 expression, but at any one time it's possible that there's only a subset of those cells that are actually competent to maintain a hematopoietic stem cell. And that, so going forward, one of the questions to address is there heterogeneity among the leptin receptor positive cells in terms of their competence at any one time to sustain a hematopoietic stem cell, and what mechanisms determine that?
Thank you, Sean. I have a second question which I think can be addressed, well, can be sent to both Sean and Simone actually, and this is about oxygen levels. So it has generally been thought that the HSC niche is hypoxic. So does it mean that there is little or no blood flowing through the HSC niche supporting sinusoids?
Simone, do you want to go first on that?
Well, I think, yeah, I think that the evidence is not conclusive about which niches are more hypoxic or less hypoxic for hematopoietic stem cells. In general, I think that most measurements have been taken in the blood vessel rather than in the parenchyma, and also whether the actual levels in the hematopoietic stem cells have A or B consequences. I think is unclear. It's very clear that hematopoietic stem cells somehow are hypoxic, and that the most primitive cells are maintaining this hypoxic state. But I don't think there is a final evidence about which particular niches are where these hypoxic HSCs reside. I don't know whether Sean has a different view on that.
I think the best data in this area come from Charles Lynn and David Skadden. Charles Lynn developed a nanoprobe that can be injected intravenously and used to image oxygen tensions in different microenvironments. What he concluded is that all of the bone marrow is hypoxic, first of all, that but within the bone marrow, the endosteum is less hypoxic. There's a higher oxygen tension at the endosteum where the osteoblasts are located because there's more arterial blood supply there. That the most hypoxic environments in the bone marrow are around the sinusoids. And even though these are blood vessels, that makes sense because not only do the sinusoids carry venous circulation, but they're carrying slow venous circulation that's not designed to transport oxygen and red blood cells around the body. The sinusoids are designed as a site where hematopoietic cells can extravasate, can go slowly through the blood vessels and extravasate out of the circulation into the bone marrow.
Brilliant, thank you. I have another question about leptin receptor positive bone marrow cells and whether they are affected by irradiation, whether their number changes or whether their localization changes after irradiation.
Yeah, we're in the process of studying that now, and what I can tell you from our unpublished data is that as Shaheen Rafi has published, the sinusoids are obliterated. I shouldn't say not completely obliterated, but severely damaged after irradiation. And so the profound depletion of hematopoietic stem cells that you see correlates with damage that you see to sinusoidal blood vessels. You see much less damage to arterioles, at least visibly after radiation. There could be other kinds of damage that's not apparent in terms of morphology. But you see a profound reduction in the frequency of leptin receptor positive cells in the bone marrow after irradiation, along with the reduction in hematopoietic stem cells. And in the 2 to 4 weeks that it takes for hematopoiesis to regenerate within the bone marrow and for hematopoietic stem cell frequencies to start recovering, you see a regeneration of the sinusoidal blood vessels, and you see regeneration of not completely normal, but more normal numbers of leptin receptor positive cells.
Wonderful, thank you. The other question I have, I guess, is fairly similar. It's about whether HSC niches change in myeloablation settings in the transgenic mice presented. Perhaps Sean has already answered about leptin receptor cells. Simone, you may want to add something about all your sympathetic and parasympathetic nervous system reporters.
Yeah, I think that, yes, regeneration must come somehow from blood vessels closer to the bone surface, which is the area that is actually preserved after radiation, and then there's plenty of evidence that if you transplant, also from your own work, Christina, if you transplant hematopoietic stem cells in this particular setting, they all tend to go near the bone surface. So I guess that somehow these vessels shown by Rolf Adams also to contain endothelial precursor cells that give rise to the sinusoidal vessels during development. So maybe the regeneration also starts in this in interface between both types of blood vessels.
Thank you. We have a new question that just came in. Which is this time not about how these HSC niches change with irradiation or myeloablation, but how do they change during aging? And I guess we'll have an answer for each of you.
Simone, do you want to go first?
Oh, you can go first.
All right. So, you know, we don't have very much data in the context of aging. Ralph Adams has recently published a paper making the point that there's real significant changes in the vasculature and in the bone marrow stroma composition in the bone marrow during aging. But in terms of the direct experiments that need to be done in terms of systematically examining the expression patterns of niche factors and conditionally deleting niche factors from specific cell types, nobody has done that yet because it's just an awful lot of different mice that would have to be aged. And so I think there's a lot of work that needs to be done to characterize precisely which cell populations are changing and in what ways in the context of aging.
Yes, I agree completely with that. I think that the Ralph Adams study was really interesting in terms of, correlating changes in the endothelial vessels with some of the changes observed in aging. But one has to bear in mind that most of the notch gain and loss of function studies showed parallel changes, let's say, in endothelial cell types, mesenchymal, perivascular associated cells, etc. I guess we all these experiments to dissect which changes cause what and remain to be done in the future. And as Sean pointed out, it will require new development of new strategies because it is not really easy to tear apart the contribution of different cell types.
Wonderful. Well, I see no more questions coming in at this time, so perhaps this is a good chance to close our session. I need to really thank hugely ISEH for organizing this webinar, and both Sean Morrison and Simone Mendes-Ferrer for giving some very interesting talks and really putting together a very nice picture of what we know at the moment about hematopoietic stem cell niches. And of course, I thank everybody who has been listening and all the people who sent in questions. And we look forward to the annual meeting and to the next webinar. Bye-bye.
Thanks, Simone and Christina. Bye.
Bye.
Bye-bye.